So it is just like microarrays. We can measure gene expression, we can genotype, and we can measure the location of transcription factor binding sites. We can also do that with next generation sequencing. The first application of next generation sequencing was sequencing the genome. This is called resequencing many times, where you have a new sample, a new human, and you want to know that person's genome. Now, but because you know it's very similar to the original reference, you don't actually do the whole thing. You stick to the places where they're different. So that permits SNP discovery and genotyping, variant discovery and quantification, transcription factor binding sites, gene expression, and also other epigenetic outcomes like measuring methylation. So it's a very widely used. This technology is used by the 1,000 Genomes Project to do resequencing of thousands, more than 1,000 different individuals. It's used by the Human Epigenome Project to measure different epigenetic outcomes, and it's used in several other projects as well. So now we described the different applications, but we didn't. Now we're going to explain how you go from this series of reads. How do we go from that to actually getting measurement from the outcomes that we care about? So we have these sequences. Now what do we do with it? So almost always the first step in when you're analyzing next generation sequencing is to determine for each read, you're going to try to figure out where from the genome this particular read came from. Right? And then we're going to use that information to do a lot of other stuff. So you have these reads here. They're actually coming from a general same area of the genome. I've selected this for the example. So you can see down here that at the bottom of the slide, you can see the human genome. And now all the reads have been mapped, is the word we use, to the genome. So they're all matched. The location of the letters are matched so that they match the reference genome at the bottom. So how is this helpful? Well, now I'm going to show you 3 examples similar to the ones I showed in a previous module for microarrays. So the first application I'm going to describe is variant detection. So with microarrays, we described genotyping and how we can use microarrays to genotype, to figure out what genotypes at each SNP a specific individual has. But with next generation sequencing, we can go further. We can actually find new SNPs. So the way it works is you take a sample of DNA, you sequence it, you align it to the genome, and once you've aligned, you can go to any specific location. For example, we can go to this location here, where this G is, and we can ask ourselves, is that a location of a SNP? And we can see that there's G's and A's. There's 2 sequences. There's a C in there as well. And that is evidence for this being a SNP. Right? We don't know for sure, but it's evidence for that being a SNP. On the other hand, if you take this C, every single read that mapped there is a C, so less evidence for that being a SNP, probably not. But here you have from the same individual, you have both G's and A's, which probably means that this person is heterozygote at that location. So not only do we find a new SNP, if we didn't know about it, but we can also genotype. There's also a C there. That's just to remind us that there's also sequencing errors, and that's probably what that is. Over here, there's another example where we see a deletion, where you can see that some of the reads have A's and others have nothing, probably signifying that one of the alleles is missing a base there. All right, so another application is RNA-seq, which is what we call when we use next generation sequencing to quantify gene expression. So how does that work? So we have RNA. In this case, we have 2 samples. We're actually going to try to find out if they're different. And now, instead of using hybridization, what we do is we take the RNA, we turn it into DNA. There's a technique for doing that, and we sequence that. So now all the RNA transcripts have been sequenced. Now we map them, and if we're interested in a specific gene, here I'm showing you gene 1 as an example. We align all the reads, and then we find for gene 1 that in sample A, we get a lot of the sequences mapping to that location, while for sample B, we don't see as many. So from here we would probably say there's evidence that in this particular comparison, this gene has more transcripts, there's more gene expression on sample A compared to sample B. And then the third application is ChIP-seq, which is what we call when we try to find things like transcription factor binding sites on the genome. And what we do, it's similar to what we explained for microarrays. We take the DNA, we have a technique that will separate out the fragments of the DNA that were bound to a specific protein. So now we take those, we take just that part of the DNA, of the fragmented DNA, and we sequence that. So we have sequenced only the sections of the genome that were bound to that protein. So now if we find a location on the genome that maps to many, many reads, that's probably evidence for that site having been bound to that particular protein. So in ChIP-seq, what we do is we align, we count for every base how many reads fall on it, and then we use these techniques called peak detectors to try to identify what locations have enough reads to make us believe that there was a protein bound to that location. So those are just 3 of the many applications of next-generation sequencing. We're going to be describing them in this class, and we're going to be learning how to turn these reads into meaningful summary statistics and how to extract knowledge out of this data.